Hereby I open this academic ceremony in which Mr. Moshi will defend his academic thesis, Epigenetic Changes During Normal and Malignant Differentiation in the Uterine Cervix. And I welcome the candidate and the paranymph and lots of success, of course. And I welcome also the promotion team, Professor Ramaaks and Dr. Holtman, and of course everybody here on site present and online. Everybody will see how it goes. And then obviously the members of the opposition committee, which you will meet later on. But before we start, we will give you the opportunity to tell us in short what you have done during your PhD training.
Yes. Thank you. Highly esteemed pro-rector, opponent, supervisor, family, and friend. In next 15 minutes, I will present the introduction, some result, and conclusion of my PhD research entitled Epigenetic Changes During Normal and Malignant Differentiation in the Uterine Cervix. Let me start my presentation with the aim of the study. The aim of the project, as described in the thesis, was to study the molecular alteration that occur in the development of different precancerous stages of cervical cancer, focusing on the so-called transformation zone where such lesions originate. In this presentation, I will focus on several items, including molecular alteration, precancerous stages, cervical cancer, and transformation zone. For this reason, I have divided my talk to 3 parts. First, I will have short introduction about cell biology of the uterine cervix. Then I will briefly present some results, which include switches on SOX protein expression during cancer development in cervix, and epigenetic changes during cancer development. In the end, I will provide you the main conclusion. I will start my introduction with anatomical overview for adult female reproductive system, including uterus, ovary, fallopian tube, vagina, and cervix. It's well established that the lower part of cervix, cancer can originate resulting from viral infection. Therefore, my research focused in this area of cervix, in particular area that has different cell type, which is called transformation zone. This slide shows you the higher magnification of transformation zone, which have different major type of epithelium, including columnar epithelium and squamous epithelium. Columnar epithelium meets squamous epithelium in the area called squamo-columnar junction. As a result of external and internal factor, transition between columnar epithelium and squamous epithelium occur during life, resulting in metaplasia and cancer in this area of transformation zone. Transformation zone has different progenitor cells, such as cuboidal cells, reserve cells, and basal and parabasal cells in the squamous epithelium. Hormonal influence or infecting by microorganism, trauma, and pH variation can induce the transition between squamous epithelium and metaplasia, resulting for immature and mature squamous metaplasia. Infection by human papillomavirus, HPV, can induce or develop precancerous lesion, which in the end lead to invasive cancer. 99% from cervical cancer resulting from infection of HPV. HPV can hit columnar epithelium, which lead to pre-malignant lesion named as adenocarcinoma in situ, which end to develop cancer called adenocarcinoma. Also, HPV can infect the basal cells in the squamous epithelium, which lead to develop pre-malignant lesion or precancerous lesion called as cervical intraepithelial neoplasia, which is known as ICE. And here it's as sin. And then develop as cancer called squamous cell carcinoma. Another possibility to form eyes and sin, HPV hit cuboidal cells or reserve cells, which lead to, in the end, to develop cancer. The question now, after I finish my introduction, I will focus to study SOX2 and SOX17. The question, why did I study SOX2 and SOX17 in cervix? For several reasons. SOX2 and SOX17 protein expression are related to normal fetal development, maintenance of adult organs, and development of different types of cancer. Also, between 2010 and 2018, before I start my PhD, few publications mentioned that these two markers are involved in cervix. This triggered us to study these markers in detail in normal and precancerous stages and lesion of cervical cancer. Then I will start showing you the result by showing how SOX2 and SOX17 in normal situation. SOX2 is a negative in columnar epithelium, and SOX17 is a positive in the columnar epithelium. While the opposite is true for squamous epithelium, that SOX2 is a positive for squamous epithelium, and SOX17 is a negative. And as I mentioned it before, that the area that columnar epithelium meets squamous epithelium area called squamocolumnar junction. At squamous columnar junction, the transition between SOX2 and SOX17 is clear. Also, I mentioned in one of the previous slides that one of the progenitor cells is basal and parabasal in the squamous epithelium. In this slide, show you that basal and parabasal is SOX17 or SOX2 positive. So the question now, what about other progenitor cells, which is reserve cells? Reserve cells, as indicated by other marker, show that SOX17 positive and SOX2 negative. Interestingly, immature metaplasia express SOX17, which is similar to reserve cells, which is SOX17 positive. This proves the assumption that reserve cells is the progenitor for immature metaplasia. Upon differentiation of immature metaplasia to mature metaplasia, SOX17 switches to SOX2, as you can see here. To conclude that SOX2 and SOX17 are differently expressed in squamous and columnar epithelium, reserve cells that contain SOX17 are the progenitor for immature metaplasia. Switches between SOX17 and SOX2 occur during maturation of metaplasia. The clinical impact of my result that the pathologist can use SOX17 to distinguish between immature metaplasia and low-grade sin lesion, because all sin lesions show SOX2 positive and SOX17 negative. And it was found that SOX2 is positive for sin 1 and also positive for sin 2. But SOX2 and sin 3 show a specific distribution pattern which could be like a diagnostically relevant. This is because we have classified the SOX2 distribution pattern to 3 patterns. Interesting observation was made in other premalignant lesion, which is eyes. As I mentioned in the previous slide, that eyes came from infected columnar epithelium. And indeed, we show that SOX17 is positive in eyes. But in few cases, we have seen that SOX17 is negative. We could correlate that the loss of SOX17 expression with epigenetic switches that occur in SOX17 gene expression. And this result is confirmed by PCR technique. The conclusion from previous result that eyes loss of SOX17 protein expression is caused by methylation of SOX17 gene. Different SOX2 pattern can be recognized. The clinical impact. This SOX2 pattern can help the pathologist to distinguish low from high-grade cell lesion. In one of the previous slides, I mentioned methylation. Methylation, I would like to study methylation in this part. So to know what's the methylation process and how does it influence the protein expression. Methylation considered as one of the major processes for epigenetic regulation in DNA activity. Methylation can turn off several genes, specific genes, and can also stop expression of the protein. These genes can be activated again by a process which is called hydroxymethylation. When it's reactivated, now that re-express the protein again. So we have studied the epigenetic life cycle of methylation and hydroxymethylation in normal and pre-malignant region of cervix. So this slide shows you the normal columnar epithelium, which hydroxymethylation is very high, while in eyes, hydroxymethylation level is really low. On the other hand, methylation, as indicated in red, is very high in eyes. The methylation and hydroxymethylation in cell lesion have a different story. This slide shows you the complicity of methylation and hydroxymethylation in cell lesion. But the take-home message from this slide that hydroxymethylation is reduced in high grade of cell lesion, which is similar to what I have shown you in eyes lesion. This led us to conclude that loss of overall hydroxymethylation is associated with high-grade pre-malignant lesion of the cervix. The clinical impact, detection of loss of hydroxymethylation could become a diagnostic marker for cervical pre-malignancies. It may be evident that in my presentation, I have mainly applied a histomorphological approach in my study. This approach, it's helped me to correlate between epigenetic switches and correlate them to understand this and this epigenetic switches in individual cells and cell population in cervical tissue sample in normal and pre-malignant lesions. This is the reason why in my last proposition I stated, seeing is believing, in particular in molecular biology. And thank you for your attention, and I will give the word back to the prorector.
Thank you, Mr. Moshi, for a very nice presentation. We're now going to start the opposition, and the first opponent is also the chair of your assessment committee, that's Professor Speel, and he's professor of molecular oncopathology at the Department of Pathology in the Maastricht University Medical Center. Professor Speel.
Thank you very much, Prorector. Dear candidate, I very much enjoyed your talk and reading the thesis. And especially I would like to compliment also your very nice images that you provided, which I know comes also from your help of your supervisors, which were actually also my supervisors. Yeah. So it's a coincidence, I think. And so I also would like to congratulate the team and also congratulate you with this scientifically interesting material in your thesis. Although I have to admit that it is, of course, about a relatively small number of cases. A lot of areas in these cases, yeah, but not so many cases. So I have always the idea you should be a bit careful maybe by concluding a lot on not too many cases. But anyway, I would like to have a question on LOH analysis, and maybe one of your paranymphs can read proposition number 3.
In cervical adenocarcinoma in situ, or ICE, a direct correlation exists between the topographical distribution of SOX17 protein expression and the methylation status of its gene promoter, explaining the heterogeneity of SOX17 immunostaining.
Thank you. So it means actually that you're saying, okay, when there is loss of SOX17 expression or no expression of SOX17, then maybe there's LOH analysis.
Yeah.
And then when I then, and particularly that's in the adenocarcinoma,
Yes.
in the columnar.
Yeah, in eyes and adenocarcinoma.
Yeah, because when I look at table 2 of chapter 2, page 28, there you also compare this expression with LOH analysis. And then when I look in the cell part, I see that about half of the cases show an LOH, and then half of the cases do not.
And the last part of the table, you mean?
Yes.
Cell
Yeah, no, with
coexistent with
methylation, the SOX17 methylation in the lesion. So you see there is no expression, but there is, in half of the cases, there is LOH analysis, and the other half there is not. So can you maybe tell me why it is different than in squamous?
Okay. First of all, thank you. Highest point. Thank you for your comment and questions. So, yes, indeed, we seen that loss of SOX17 expression is correlated with the methylation, and it's known that HBV is integrated in all cases of ICE, and it has been published that 20% of SYN lesion, especially SYN3, it's correlated with the HBV integrated. So we were thinking that the methylation of SOX17, for example, and the high grade lesion of SYN3, it's correlated with the HBV integration.
Okay, that's an interesting link, but that means then that in half of the cases, although there is no expression, LOH, and there is LOH, it does not. So LOH is not always in the squamous epithelial linked to no expression in that sense.
Yeah, we haven't seen any expression of SOX17 in squamous epithelium.
Yeah, but sometimes there's LOH, sometimes there's not. So
yeah, it could be there is some expression for that, but we haven't seen it in our case. There is no expression for SOX17, but we saw methylation level of methylation level of SOX17 gene expression, but it's not directly linked to loss of SOX17 expression in normal squamous and SYN.
Could there maybe be another? I mean, is it the only epigenetic mechanism maybe that could be linked to that?
SOX17 is an important factor in Wnt signaling pathway. It could be it's blocked by any marker during Wnt signaling pathway. And in it has mentioned that beta-catenin can block the SOX17 in colon cancer. But in our case, we haven't seen any expression for SOX17 in normal and pre-malignant lesion. I mean, SYN.
You didn't look at beta-catenin in that sense?
No, we haven't came across the testing beta-catenin, but could be for future work and could be one of the suggestions for working after.
Maybe another, because you introduced it in fact yourself already by discussing HPV integration.
Yeah.
And you have tested that with in situ hybridization, sometimes with fish, sometimes with kish. Is there a reason why you use fish or kish, or?
Like to see the, we use fish and kish to identify the viral load of HBV.
Is that more easy with fish or with kish? Or?
Both will give you a similar, just the difference is using a microscope.
Okay,
Yeah.
so it's more
fluorescent
like
microscope or bright field microscope.
Okay.
It's
And
a fluorochrome labeled peroxidase.
Okay. But you said maybe this loss of SOC17 could lead to integration, or does that has to do more with this, let's say, how do you say it? The loss of this 5-hydroxymethylcytosine, which you sometimes see.
Yeah.
What can you say something more about the mechanism? How
You mean in SEN?
Yeah, if you say, well, this maybe can lead to
Yep.
integration, I mean, how should I see this?
Yeah, we have found that the integration in 20% of SEN lesion.
Mm-hmm.
And the loss of hydroxymethylation or 5-hydroxymethylcytosine, we have seen it in 20% of cases that in SEN3.
These were the same cases then?
No, it's not the same case.
Not the same cases.
Not the same cases, but we are thinking that could be linked between integration of HBV and also loss of hydroxymethylcytosine. Also, another point that
and why? Can you elaborate something more on why could that be a link?
Yeah, because when HBV integrate the human genome, that could mutate the TET, 10-11 translocate. That the TET enzyme is important to repair the still replication fork, which is important to integrate HBV in human genome. When it's TET mutated, it will not fix the replication fork. Mm-hmm. But if it's mutated, you will not have ability to fix this replication fork, and then
and that maybe can lead to breaks, or
it will lead to integrate the HBV, and it will be reduction of hydroxymethylcytosine.
Thank you. I think the pro-rector would like to give others the room. Okay, thank you very much for your answer.
Thank you, Professor Speel, for your position. Then now we come to a following member of the assessment committee, and that's Professor Steenbergen, who is Professor of Experimental Pathology at the Amsterdam University Medical Center. And thank you, Professor Steenbergen, for coming all the way from Amsterdam, albeit online, but we are always pleased with guests from outside.
Thank you, Mr. Rector. And dear candidates, I'm really honored to also discuss your thesis with you. And first of all, I would like to congratulate you, but also your promotion team, because I really enjoyed reading your thesis. And I think the images, as was also mentioned by the previous opponent, they are really wonderful. And I really like the fact that you really went into depth into the figures to see what's really going on at the single cell level. My first question was on why did you choose to use SOX2 and SOX17, but you actually just answered it in your presentation. So I'll just go to my second question, which relates to that, because during your PhD trajectory, the paper came out by Chumduri in Nature Cell Biology. I don't know whether you can see it on the screen. You probably know the paper. And they are really doing a similar investigation as you were doing. So can you tell me how your findings on SOX2 and SOX17 relate to their findings in their paper? How they relate to, like, the Wnt pathway?
Hi, STEM opponent. Thank you for your question and comment. So this paper, one of the a recent paper, I think in 2021. And they mentioned the transition between different marker and squamocolumnar junction. And this paper emphasizes more about the microenvironment from the stroma. So they see that this microenvironment, which is guided by Wnt signaling pathway, can induce marker and can inhibit markers. So, and indeed, we have, like mentioned, that SOX2 and SOX17 is an important marker in Wnt signaling pathway. But maybe like it's wise, for example, to combine or to, they should, for example, they should see how SOX2 and SOX17 affecting by microenvironment. Is the microenvironment, for example, induce formation of immature metaplasia by affecting the reserve cells? That immature metaplasia is express SOX17, for example. It could be induced by microenvironment in the stroma or not. And also when signaling.
Yes, I was. So indeed, they related to the expression in the stroma. So did you? Because I tried to look in your figures at the stainings for SOX2 and SOX17 in the stroma. I couldn't really figure out whether there was a difference for those proteins also in the stroma. Did you look at that as well?
We haven't seen express of SOX2 and SOX17 in the stroma at all.
Not at all. I thought I saw some, but that's fine. So, but am I correct saying that like SOX2 is inhibiting Wnt and by that directing the cells into the squamous differentiation direction?
No, SOX17 inhibited by beta-catenin. And both SOX2 and SOX17 is a tumor suppressor gene, and it's also in Wnt signaling pathway, under umbrella of Wnt signaling pathway. But SOX2 is.
So you say it's tumor suppressor. I think people also propose SOX2 being an oncogene. Is that incorrect?
From my knowledge, no, but I for sure like SOX2, it's a tumor suppressor gene because it's inactivated to become a cancer. But oncogene, it's mean that it's a gene that could be activated abnormally to cause a cancer. But SOX2 is tumor suppressor gene.
But still you see like an overexpression of SOX2 in the SYN3 lesions in the cancer, and you see a gain of the chromosomal location. Would that not indicate in the direction of an oncogene?
Can you repeat the question, please?
So you see a gain of 3q26 where SOX2 is located in SYN3 lesions and in cancers, and you see an overexpression in SYN3 lesions and in squamous cell carcinomas. Would that not indicate that it's an oncogene?
It's we have we have seen that SOX2 is increased with the progression, and also we several publications they link the expression with copy number of SOX of chromosome 3, and it's chromosome 3 it's amplified or gained during progression of the SYN and increasing SOX2 from in SYN1 and SYN2 and SYN3 was observed in our cases, and also it's confirmed that maybe link of SOX2 expression with the copy number is also it's confirmed in expression of SOX2 in BETN3 when we see like kind of disomy of SOX2 gene, and but SOX2 was negative.
Okay. I think because of the time, I would like to go to one next question if time still is available.
Short question.
And I would like to. Okay, so if you go to page 80, there you show a really nice figure of an adeno lesion and a SYN lesion, and maybe you can just answer by yes or no. Are those two lesions derived from the same progenitor cell or from different progenitor cells?
Yeah, I think that I cannot answer it yes or no, but I need to elaborate. But here, like, maybe it came from one. Yeah, maybe it come from different from one progenitor cells, which is reserve cells, And we have seen different marker, for example, SOX2. For if SOX2 is positive in eyes, we link the positivity of SOX2 in eyes for two reasons. Maybe one of the reasons because, as I mentioned, it's increase of the copy number in the pre-malignant lesion, and also maybe it was like kind of a genetic mistakes that eyes producing SOX2, which is positive, because they share the same reserve cells. It's kind of bidirection reserve cells.
Okay, I think because of the time, I probably should stop. I would be very interested at some point maybe to also look at HPV and the HPV type and variants in those two regions, whether they are really the same or not. Maybe that can also answer your questions in the future.
Thank you, Professor Steenbergen, for your opposition. Now the following member of the opposition is also a member of the assessment committee. That's Professor Beckers. He's professor in prevention of gynecological cancers, in particular pre-malignant lesions of the cervix at the Department of Obstetrics and Gynecology in the Maastricht University Medical Center. Professor Beckers.
Thank you, Pro-Rector. Also, I would like to start with congratulating you with the nice work.
Thank you.
Also, the pictures appear to me very nice, so I also want to congratulate your team in this success. I would like to start with asking one of the paranames to read proposition 5.
Women with cervical cancer are overtreated.
Thank you. In general, I would agree that women with cervical cancer are overtreated, but I think you want to say this is women with cervical neoplasia, including all the pre-malignancies, because you don't talk too much about cervical cancer in your thesis, but you talk a lot about pre-malignancies.
Yeah.
Can you tell me how this relates to the results of your thesis?
Okay. First of all, thank you for high SLAM opponent. Thank you for your comments and the question. By answering this question, first of all, I want to draw your attention. I am not a gynecologist. I cannot say treat this patient or treat. But I make this conclusion for several reasons. For example, when pathologists receive a biopsy, they see it, and if they see reactive lesion or reactive area, they consider it's high grade or low grade. But our marker, for example, SOX17, by staining this biopsy or this section, can give you an idea. This is not a lesion. It's immature metaplasia. And one of the, like, for example, pathologists will decide, okay, remove this area. What will be harmful for women to remove its normal area as nothing happened. But another reason, maybe it's overtreated if, like, pathologists decide to remove or to suggest to remove low-grade lesion. But I feel like you should follow wait-and-see strategy, because all SIN1, I won't say, most of SIN1 is going to regression. Yeah. Because immune system will enhance in the infected area, and that will be clear the HPV. Okay. Another.
No, thank you. I understand you know a lot about this subject,
okay.
So I will want to move on to the next subject. I would like to go to page 31, figure 6, and I was wondering about 6C especially. If you look at page 31.
Okay, 31.
It's figure 6. And the first, in A, you show normal squamous epithelium and normal columnar epithelium and the original squamous columnar junction.
Yeah.
I understand it. In B as well, you see columnar epithelium, squamous epithelium, and the immature metaplasia in between. I understand that as well. But then in C, you put squamous intraepithelial lesion, and you put it in between the immature metaplasia and the columnar epithelium, which I think, why did you place it on that spot? Why did you not put it in between squamous epithelium and the immature metaplasia? Because it looks now like if you would think this is a timeline, then you think, oh, it's columnar epithelium, and then SOX2 becomes positive, and SOX17 becomes negative in squamous epithelium, and then it goes on to form immature metaplasia, and then it switches back again, and then it switches back again to squamous epithelium. So that's why I thought this doesn't seem logic to put it in a picture like this, but...
Okay, thank you for your question. So we, in our result, we indicate that when the HPV infect the immature metaplasia, the lesion goes directly to SYN2 and SYN3. It's high-grade lesion. So because of that, we bought like a immature metaplasia and then cell. It's not like it's a new squamous columnar junction. And SOX2 positive and SOX17 negative, that we call it old squamous columnar junction.
Yeah, but then I would suggest that instead of putting the blocks next to each other, you put them under each other, because there are two ways. It can go directly to squamous intraepithelial lesion, or it can become immature metaplasia. So then you would for me would be more logic.
Okay, thank you for the suggestion.
The next question about this is if the switch takes place from SOX17 positivity to SOX17 negativity and SOX2 negativity to SOX2 positivity, so it goes to the squamous epithelial phase. Is there a way it can switch back as well, or is it only one direction?
So there is a way. It's called our case is squamous columnar junction, or which consider metaplasia. Metaplasia, it's columnar epithelium replaced by squamous epithelium. There is no during differentiation, but maybe there is a metaplasia by transdifferentiation from squamous epithelium to columnar epithelium, but this case is not in cervix. Okay. This is in the between esophagus and stomach.
Okay. So in the cervix, it's only one direction.
Only one way.
Okay. Well, given the time, thank you for your answers. I'm very satisfied with that.
Thank you.
I'll give the word back to the prorector.
Thank you, Professor Beckers, for your opposition. Now we come to the following opponent, and that's Professor Moret. He's professor in host pathogen genomics and public health in the Department of Genetics and Cell Pathology in the University of Maastricht. Professor Moret.
I would like to thank the pro-rector for the opportunity to discuss the thesis with the PhD candidate, but first I would like to congratulate the candidate with a very nice thesis. Also want to congratulate the PhD team consisting of Professor Frans Ramakers and Dr. Ton Hopman. And finally, I want to congratulate you and your family, friends and colleagues who have all contributed in their own way to make it possible to defend your thesis today. Not many will know it that you tried, and especially together with Dr. Elena Ambrosina, to obtain a PhD position at the Institute of Public Health Genomics at our Department of Genetics and Cell Biology. Unfortunately, the Yazan University was not able to provide you funding for that. That is really a pity. But I congratulate you with the fact that you have obtained a position at the Yazan University. It's very nice. And the fact that Elena and myself are both here should not be seen as revenge that we will make the questions very difficult. We will make it nicely today. As you know, I like immunogenetics of infectious diseases. Many infectious diseases, for 30 to even 50%, are based on how your genes look like, how you combat infections, how your immune system recognizes and goes with them. As you perhaps know, one of my PhD students, Ivan Brankovic, made a review on cervical carcinogenetics and the gene polymorphisms, or single nucleotide polymorphisms, in 2017. It's not your expertise, but could you, based on your knowledge on cervical cancer immunology, contemplate which genes could be relevant and which pathways could be relevant if you look to immunogenetics? What kind of genes would you think could have mutations that will abolish their functions and be involved in cervical carcinogenesis?
from the 2 hour markers, SOX2 or SOX17, you mean, or from?
For instance, but perhaps even broader, on how the immunology of an infection works, what kind of genes are involved in there. It's a complex question, I know, but I want to try to see if you can have a discussion around this.
First of all, thank you so much for comments and the question. So if I have to choose SOX2 or SOX17, I will choose SOX2 because for several reasons. SOX2, it's, as I mentioned previously, that SOX2 it's located in chromosome 3, 3q26 area, which is amplified during progression of cervical cancer. That amplifying of the progression can involve in several problems, and it could be induce several issues, for example, like suppression. It could be after integrated HPV in human genome can implement another issue for growing for cell proliferation and cancer growth. And also SOX2, I choose SOX2 also as an important factor or important gene in Wnt signaling pathway.
I will interrupt you briefly because you're almost answering my second question. In 2022, Vajada et al. Published that SOX2 is known to misregulate in cancer cells by changes in microRNA function due to SNPs in actually the binding sites. So your comment on if you have to choose, take SOC2, it's a very good one. People have done it and have actually found SNPs in that gene. And SOC17 is not yet studied in detail for that. So that makes sense. I want to go back one small step. If a cell is infected with a microorganism like HPV, what kind of immunological reactions will that cell potentially take? Because it's a hostile environment where there is a microorganism which should not be there naturally.
Microorganism, HPV or another microorganism like a microbiota?
Maybe HPV, but may, for instance, also be chlamydia, for instance. What would a cell do?
Like first for chlamydia or streptococcus, I think the innate cells, it will be the first line to defend or to get clear from this infection. Innate immune system.
Yes.
Yeah.
So, for instance, cytokines and chemokines.
Yes.
So, these kind of cells have their natural immune response, indeed, making cytokines, chemokines, negative regulators of inflammation, maybe toll-like receptors who regularize microorganisms. All those genes, they have mutations in them. And those mutations make those genes function better or actually lesser. So it's a way of looking at infection and using that for markers for making the diagnostics and assess how well people will combat infections. If I look to your summary of discussion and to your valorization chapter, I hope that based on your figure 1, you would make some kind of integrated model on how to diagnose cervical cancer. So if you now can choose, what should be the optimal variables you should do if you want to take a vaginal swab or do something else? What kind of molecular or immunohistochemical or any other ways would be the perfect way to diagnose women, taking into account also your findings inside your thesis?
Thank you for your question. And as I mentioned before, I'm not pathologist to classify or to diagnostic. And I will suggest how I will use the markers in my thesis to diagnose the samples. So first I will choose, for example, SOX2 and SOX17 to see if this area is a normal or not. If it's normal, like it will express SOX17, is immature metaplasia, or if it's normal that express SOX2, and it's not normal, it will express SOX2 in sin lesion. But also I will get help from methylation and hydroxymethylation, because we have seen that hydroxymethylation is reduced in high grade of premalignancy of eyes and sin lesion.
Yeah, I think. Thank you very much for your answers, but I saw the prorector and my time is finished. Thank you very much.
Thank you very much, Professor Moret, for your position. Now we come to the following opponent, and that is Dr. Ambrosino. She's Associate Professor of Immunology and Reproductive Tract Microbiota in the Department of Genetics and Cell Biology in the University of Maastricht. Dr. Ambrosino.
Thank you, Mr. Prorector. Dear candidate, congratulations on your work and a nice presentation. Congratulations to the supervisory team as well. I very much enjoyed reading your thesis. So in the first question, I would like to discuss with you the potential role or association with the microbiota, so the commensals. So we know that the commensal bacteria in the cervicovaginal area, the so-called cervicovaginal microbiota, seem to contribute to immune defenses against and also hinder colonization by pathogenic microorganisms. In the case of HPV, it has been reported that an abnormal cervicovaginal microbiota, so the dysbiotic states, are associated with infection by the virus and also with different progression stages of cancer. So my question to you is whether you think that the cervicovaginal microbiota play a role in the biological pathways you described. If so, whether you can think and try to explain the association of those pathways with the microbiota.
First of all, thank you so much for the questions and comment. So it's outside of my scope, but I will say it will influence. For example, a normal microbiota, we have it in our body. For example, lactobacillus, it exists in cervix. But on another microbiota with, for example, Synethia, is associated with the infected with HPV and cervical cancer. So the Synethia and another chlamydia that can increase the lactic acid and H2O2, which lead to increase the pathogen atmosphere, and then also it's increased for cytokines and pro-inflammatory cytokines. If they are working together, HPV and, for example, microbiota, I think yes, because it has been published that, for example, antibiotic that can treat the chlamydia, that can induce immune system. If the woman has a low grade, it helps the woman to cleanse the HPV. But this antibiotic will not help to get rid of the HPV if the woman is in SIN 3 or SIN 2.
So, but would you expect, that the different status of the microbiota will be associated, for example, with regulation of the transcription factors that you investigated?
We haven't tested, but maybe it's good suggestion for future if we can link the influence of microbiota with HPV for SOX2 or SOX17. I don't have any idea because we haven't tested yet, but I'm sure it will affect.
All right. You might need different types of samples, but it will be interesting to investigate. The second question, I would like to go back to the global relevance of your work. So as highlighted in your thesis, I'm quoting from it, cervical cancer is still the 4th most frequent cancer in women in developing countries due to the absence or limited accessibility to population screening and HPV vaccination programs. So in light of this burden, also disproportional burden in certain regions of the world, how do you envision your results to practically contribute in addressing this challenge in those regions?
So our SOX2 and SOX17 marker, it will add extra marker for pathologists to they have a panel of markers, but it will help to, for example, to get a right classification for SIN1, SIN2, or hematoma metaplasia. But with for example, low and middle income country, I think the marker will not have a significant impact on there. They need screening, they need vaccination first, because I think screening and vaccination is much cheaper than our markers.
Yeah, and the impact probably will be much bigger at the beginning to,
yes,
to tame this burden. And so, but you would expect that such marker would be applicable in different settings anyways and on different populations?
Yes. Yeah.
All right. Looking forward to investigating that.
Thank you.
Thank you.
Thank you, Dr. Ambrosino, for your position. And now we go to the following opponent, and that's Professor de Kock, who's professor in population-based toxicogenomics in the Department of Toxicogenomics in the University of Maastricht. And today he has also accepted the role of secretary of this committee, for which I'm very grateful. The floor is yours.
My pleasure. Thanks. Congratulations on this extensive piece of work. I must say this is beyond any doubt the thesis with the largest number of colorful pictures I've ever looked at. And looking is not good enough, but seeing is believing. So the added value of understanding added to the looking is the seeing. And that requires a specific skill which you apparently possess, and I'm still figuring out if I do that too. So I have a number of questions on that later on. But first I would like to go to chapter 6, where you look at methylation and hydroxymethylation analysis. And I have two short technical questions. One relates to the fact that you're using formalin-fixed and paraffin-embedded material. And we also use in our toxicological studies that type of material to extract nucleic acids, typically for transcriptome analysis, but also for epigenetics. And then we find that this method of preserving has an impact on the integrity of the material, and due to particularly oxidation processes, it degrades depending on the type of methodology. So I was wondering, did you investigate if the formalin fixation actually has a capacity to oxidize or maybe even dehydroxylate your 5-hydroxymethyl cytosine? So the question is, are you sure you're not looking at some sort of a potential artifact?
First of all, thank you for your comments and question. So, unfortunately, we haven't checked about if formalin will have effect or have any influence in hydroxymethylation or not. But in the chapter 5, we have this different pretreatment, retrieval method. So all these retrieval method will have a kind of impact on accessibility for DNA and the compactness of the DNA itself. But for hydroxymethylation, I don't think, we haven't tested.
Okay, thank you for that honest answer. Maybe it's something to look into the future. A second technical question is related to the semi-quantitative analysis. So you do a visual interpretation of the data, and I was wondering if there's somehow a standardization step, or did you do any type of cross-validation to make it less researcher independent?
In which chapter?
To be exactly on page 129. You mention, you write, we classified the immunofluorescence intensities as negative, weak, intermediate, or strong. Now that's semi-quantitative.
In 129?
On page 129. But I think it's in general to the assessment of these type of images. So there is a sort of a judgment. How reproducible is that quantification? Because there must be certain transitions or classifications that someone else might classify a little bit different.
Yeah, this is the seeing is believing. It's what we see under microscope.
Believing is what we do in church or something, but this is in academia.
Yeah, but so we make this classification depend of our observation. Like, for example, we say both negative, we number it, for example, number 1, one positive, 1 negative, number 2, and number 3 if we have seen both positive. But also the intensity different, we number it and depend of the observation. Maybe, as you said, everyone can have a different classification or but in general, the results show that there is a kind of reduction of hydroxymethylation.
Okay. So did you ever consider using machine learning algorithms to look at your image analysis and retrieve specific features that may even be driven by your fluorescence, but may reflect something else that we as humans cannot see, but that a machine learning algorithm can actually determine much more precisely than we can and much more reproducible in order to classify disease states?
You see the machine.
Mm-hmm.
To my knowledge, I don't know that a machine can see the different, but maybe we can check another approach. But I cannot mention any machine that it's come in my mind.
Yeah, well, that's what we do now in our disease transition states in toxicology. So we see these images and look into features that are not necessarily recognized by human vision, but by other characteristics. And as you have all these images, it's still a possibility to
but
look into.
We have seen this, the image, we took the image by fluorescent microscope, and we took it by confocal. We see the resolution and the quality of the classification and the brightness. It's for loss of, for example, hydroxymethylation, it exists there. Like,
yeah, well, I would like to move on to the next topic, because this can be a long discussion, which is about measurement of global methylation and hydroxymethylation. So there are also methods, of course, to look much more specifically into specific methylation, so not just at the promoter sites or global, but at specific promoter sites. And that would have given the opportunity to do much more specific analysis. Have you ever considered doing that?
For which?
For specific genes. So now you look at global, so it's a general hypermethylation or hydroxymethylation in certain areas. And you can make associations. Actually, it was one of your conclusion slides. You have associations, but it's, I would say, just associations. But you cannot link specific genes being silenced by methylation or prone to be demethylated as a consequence of hydroxylation. You just have the images of a global methylation pattern.
But maybe that idea, you will not have the difference between single nuclei to another. But by immunohistochemistry, can identify a single nuclei, it's positive, and the neighbor nuclei is positive. By another mechanism, or for example, multiomics or PCR or sequencing, they pull the DNA together, and you will get a result. You will not see the heterogeneous between nuclei to another nuclei.
No, that's related when it comes to cell-specific responses, but I'm now talking about the lower level of the single genes that would be involved, because you aim to unravel the mechanisms that are involved. And based on global methylation patterns, you cannot identify specific genes that were methylated or hydroxymethylated.
Yeah, so you cannot identify a specific gene that is hydroxymethylated or not. We are in this chapter, in this study, we study the overall methylation, and we compare methylation with hydroxymethylation. After we got linked that methylation, for example, methylation can link with SOX17 expression, and we decided to study the overall methylation.
You may finish your answer. You may also say enough is enough. Enough is enough. Okay.
Okay. Thank you for that response.
Thank you, Professor de Kock, for your opposition. And Mr. Moshi, the time appointed for the defense of your thesis has passed, and the degree committee will now withdraw to discuss the quality of your thesis and the way you have defended it. And I request that you and your company here await the results of our deliberations and our return, and also for our guests online. We will be back.
Thank you.
Excuus.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side. 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Take the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you've won the battle once you've grown. South side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile.
I reopen this academic session, and Mr. Moshi, the degree committee here present has assessed the quality of your thesis and of your defense, and in view of its positive verdict and taken into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. And Professor Ramakers is authorized to confer upon you this academic distinction in accordance with Dutch university custom. And I invite you, supervisor, to now take the floor.
Mr. Jobran Moshi, do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
Then, by virtue of the authority vested in us by law, in accordance with the decision of the Defense Committee present today, I hereby grant you, Jobran Mohammed Moshi, the degree of doctor with all the rights and duties associated with this by law and custom. As evidence thereof, I hereby give you the degree certificate, signed by the rector, secretary, and other members of the Defense Committee, and bearing the Great Seal of the University. Highly esteemed Dr. Jobran Moshi.
Thank you.
It is with great pleasure, of course, and pride to be the first to congratulate you with this honor, with this achievement. It's the title of Philosophy Doctor, which is the highest academic distinction that we can provide you with. I think you will also agree that the 5 years of our research were a roller coaster, not only in a scientific way, but also in several instances, an emotional roller coaster. But finally, it has led to a beautiful thesis. Yeah. Everyone agrees on that, and to several publications. What I would like to do also is to congratulate your family, in particular your wife, Dua, and your son, Batal, your parents, brothers, and sisters. But also, I would like to acknowledge here the never-ending efforts of your daily supervisor, Dr. Hopman, Tom, during the last 5 years, and also the efforts from Monique Ummele, Mieke Henfling, and Jos Broers, all the others that have provided not only technical support, but also helped you with private matters in the past years. I wish you and your family a happy life back in Saudi Arabia, and for you personally, a lot of success in your position as assistant professor at Yazan University. I would like to give the floor now to your daily supervisor, who will present the more informal and more anecdotal part of the laudation.
Okay, thank you. Highly esteemed Dr. Jobran Moshi, dear colleague, my friend. Congratulations. France has spoken official words. For me, another side of Jobran. You wrote in the acknowledgement of your thesis, the journey of obtaining the PhD degree has finally come to an end. You said journey. Perhaps it was better to call it a survival travel. Following, let's say, Fata Morganas, lack of scientific water, and missing green oases. But with the persistence of the team and your never-ending belief, we have this also one of the opponents asked you the belief, in what you saw, we have found a way through the desert. And at the end, a beautiful green oasis. This travel is nicely described and illustrated in your thesis. Things in Maastricht were not always easy. Remember the Limburgian dog accident. A dog became aggressive and you felt in the brandnetels. So these are unknown in Saudi Arabia. So he went to first aid and ended up close to the intensive care. Yeah, but you now are familiar with it. And you escaped from being captured and 2 weeks in prison. Driving on the highway, a car was passing you and indicated in red, volg mij. What is volg mij? So Jobran, his action, bypassing the car at maximum speed. So result, wailing sirens and blue light. He was forced to leave the car and was frisked. So let's say hands on the car, legs spread. Your charm was used to explain the misunderstanding and also driving license issues. So, and the Beckman ultracentrifuge accident. A not properly closed lid of the centrifuge resulted in destabilized rotor with a total loss of the centrifuge. And lucky that the centrifuge was already 30 years old. So that was good. And you're marking the total lab with yellow post-it notes. So you help to find things back in the fridge. Luckily, Monique informed everyone properly. And okay, I'm sorry, that was for the centrifuge, because we also got a penalty from The Hague, official warning that we had broken with GGO's bacterial growth. So that was a serious accident. But Monique, she solved this. When there is no electricity in a private house in the Netherlands, not the local government are taking care of these facilities. So it's a private initiative if you have problems. And going to your car, let's say, or driving in the Netherlands in a Suzuki Alto, I think, yeah, you know that car. We are not ashamed when we drive in such a small car, but for you, driving in a black BMW has more status, and I could imagine. And I would say this also what we should remember. We also had the Corona period in your period in the thesis. So this period strongly influenced your research, also collecting material, collaborations, and visiting meetings. Jobran, we will miss you. And not only for the discussions about research, but also for the things you always took with you when you are traveling or you were celebrating. You always fed us with friendly, sweet cookies and sweets. I wish you and your family a happy life back in Saudi Arabia and a good new start.
Thank you. Thank you. Thank you so much.
Thank you, Professor Ramakers and Dr. Altman, for a beautiful laudation. Dear Dr. Moshi, also on behalf of the Maastricht University, I congratulate you with the degree you have acquired. I hope you can celebrate it today and perhaps also back in Saudi Arabia. And I also congratulate, of course, Professor Ramakers and Dr. Altman, your supervisors, and your family and friends here and in Saudi Arabia. Having said that, we come to a closure. I would like to thank the opponents for all their work, and of course Luc Peters and his assistant Anne Pedel for making this session possible on site and online. What is going to happen next is that I will ask the audience to leave this room, because we are going to make a picture here with Professor Steen on the side and Steenberg, and then we go to the stairs where we make another photo, and then we go to the reception room. So you can already go to that room, that's called the rafter, and when you are asking yourself when can we congratulate the candidate, it's quite simple. We go first and you follow us. Okay? And with that, I close this meeting.